All right, so let's continue our discussion on the single cell data analysis pipeline. Just as announcement, we have posted the first homework, which will be due in 2 weeks on Saturday. So I encourage you to start early in case you run into any problems. So following our past 2 lectures on normalization, here the key is that we want to remove the effect of cell library size. So we want the cell library size to no longer have an effect on our data. And as a recap, this is the default option in CIRAC called the size factor normalization. As a review, what it does is that first it normalizes the counts for every cell so that the total count for each cell is equal to a constant, and the constant by default is set to 10K. So it's like 10,000. And then you add 1 to the count, normalized count, and then you do log transformation. So it's called log 1p CP10K. But there's also a way to set a total count to be 1 million. And it has been observed that this total count will have an influence, right? So we have some discussion about why the default is set to this from the variance stabilization perspective. Another method included in CIRAC was called the SC transform. It's a regression-based method, but in the regression, essentially, we're trying to remove the effect of log 10 cell library size. So the take-home messages we gave at the end of last lecture is that this log 1p CP10K is CIRAC and scan by default. It's justified from the variance stabilization perspective because it's found that this is similar to the log 1p PF, which you will try in your first homework to see whether they're really similar. But there's no consensus on the best performing normalization method. It's still a big open question. So it can be a good practice for users to test different normalization methods and compare the results in your downstream analysis. So in your homework, we want to let you explore the differences between different normalization methods and whether they satisfy variance stabilization, right? And the reason why we need variance stabilization is because we want to make the data more like normally distributed, so that in the normal distribution, the mean has nothing to do with the variance. Then we can apply techniques designed for normal distribution. Okay, but before we move on to the next step, I want to introduce this very widely used technique, but not in single cell data. For bulk data, this is called quantile normalization. And quantile normalization was first developed for bulk microarray data back in 2001, and has been then used for bulk RNA-seq data, but not so much for single cell RNA-seq data. So I want you to think about why this might be the cause. What does quantile normalization do? Basically, if you have multiple samples in your bulk data, one sample means one experiment. So for example, if you measure the same biological sample for 5 times, then you will get 5 bulk samples. And for each sample, you will get a distribution of gene expression levels across genes. Okay, so it's the distribution across genes within the sample. And you can see that each line, each curve here represents one density function, one probability density, which represents the distribution. But they're not all aligned, right? But when you do the quantile normalization, this is what you will achieve. You will get just like one density curve because they're basically matched. So what does it do? So this is a very simple illustration from this paper. So in which every column is one sample, and here we're just looking at 5 genes for simplicity, and here the data are counts. So what we aim to do is that we want to make the 5 values distribution to be the same across the 4 columns. That's what we want to achieve. But then I'll ask you, why do we want to achieve this? Which we will talk about in next slide. But you can keep on thinking this. First, how do we do it? We need to create some reference distribution so that every distribution will be transformed to the reference distribution. But in this case, there's no single distribution that can be regarded as reference. The 4 columns are kind of on the equal background, right? So therefore, the first step is trying to get an average value for the smallest value, second smallest value, third smallest value, fourth smallest value, and largest smallest value for the reference distribution. I'm sorry, so we are giving lectures, so please postpone your conversation to the end of the class. So then what do we do? Within each column, we will sort the values from the smallest to the largest. So that's what we do. So you see in the first column, 2, 3, 3, 4, 5. That's the sorted order. Then 4, 8, 8, 9, 14. So we are sorting the value in each column from the smallest to the largest. Then in the first row, we take the average of the 4 smallest values. So we get 3.5. So 3.5 is just the average of these 4 numbers. 5.0 is the average of these numbers. Then we will call these values as the reference distributions values for the smallest, second smallest, third smallest, and the largest. Then what we do is that we go back to the original order. So here the authors use the colors to mark the order. But we replace the values by the average. So basically 2 becomes the smallest average, 3.5. 5, the green second smallest, this one, oh sorry, not the second smallest. 5 in the first column is the largest. Then we pick the new largest value, which is the last column, it becomes 8.5. So we just fill in these entries by the new values. That's the quantile normalization. Then you will not be surprised to see that after quantile normalization, the 4 columns have the same distribution. But it doesn't mean that every gene, every row has the same value. That's not the case. Because you see this gene 1, its values are 3.5 in the first 2 columns, but 5.0 in the last 2 columns. So that's what we'll achieve. So we will make the distributions align, but for each individual gene's values in those samples, they could still be very different. Okay, then why we want to do this? So this paper, which I found very interesting, introduced a way to help you decide whether quantile normalization is appropriate. So in short, basically the author's messages are that you could use quantile normalization for the first 2 cases, but you don't want to use it for the last case. Why is that? The intuition is for the first 2 cases, these density curves are not perfectly equal to each other, but they are largely similar. So then you would think it's reasonable to assume that, okay, the within sample gene expression distribution doesn't change much from sample to sample. Then you align these curves together. But in the last case, you actually have samples from 2 groups, brain samples and liver samples. And you can see the red curves and the green curves are very different from each other. So in this case, it doesn't make sense to assume that the gene expression distribution is the same in brain and liver. That's the intuition given. But I just want to let you be aware that given this, you can see how subjective it is to decide which normalization to use, right? You need to first assume something is unchanged. So in the previous size factor normalization, we assume the cells to have the same library size. And in the assay transformation, we assume that there is no relationship between every gene's counts and the cell library size in the negative binomial generalized linear model. So we have to make so many tricky assumptions there. And here you're assuming that the overall distribution of all genes is unchanged across samples, from sample to sample, even though for an individual gene, right, its relative value in each distribution and samples could be different. So that's why I feel like microarray or this approach has success in microarray. It must be for some reason, right? It could be that in microarray, the differences we see is largely due to some technical artifacts such as probe affinity levels, right? How likely the probe can be attached to the gene you want to measure. For those reasons, people found this successful. But then the question I want to ask for you is, why isn't it used for single-cell RNA-seq data? Because for single-cell RNA-seq data, if I want to use this approach, then each curve will be 1 cell, right? We look at all genes within each cell. I guess one possible reason is that single-cell data is so sparse. When it's so sparse, you have many genes with 0. When you have this case, then the quantile cannot be well defined, right? Because you will have a lot of zeros in each column that will create ties. That's what I think. Yeah, but you can keep on thinking this question. I just think quantum normalization is something good to know. All right, so then let's move on to the next step, identification of highly variable features or feature selection. So let's just talk about single-cell RNA-seq data. Then in this case, the features are genes. So this is what's on the Seurat tutorial. We next calculate a subset of features that exhibit high cell-to-cell variation in the dataset. That is, they are highly expressed in some cells and lowly expressed in others. We and others have found that focusing on these genes in downstream analysis helps to highlight biological signal in single-cell datasets. Our procedure in Seurat is described in detail here and improves on previous versions by directly modeling the mean variance relationship inherent in single-cell data. And it's implemented in this function, find variable features. By default, we return 2,000 features per dataset. These will be used in downstream analysis such as PCA. So you see, this is the command, find variable features, and PBMC is the Seurat object, and the default selection method is called VST, standing for variance standardization. That's my guess. And number of features by default is 2,000. So you will only retain 2,000 genes instead of all the genes after this step, right? So if you recall, in the Seurat object, the genes are rows and the cells are the columns. Okay, and this is what you can do with it. So once you have done the find variable features, then these are stored in the PBMC object. Then you can keep the top 10 genes for your examination. So specifically, given the variable gene selected, variable feature selected, there is a function for plotting called variable feature plot, which will give you something like this. The y-axis is the standardized variance. Regarding the standardization, we'll talk next. The x-axis is the average expression. Okay. And you want to find a threshold in the y-axis so that the top genes in terms of standardized variance would be selected. Okay, this is basically showing the selected genes in red, the unselected genes in black. But I want you to be aware that there might be some overplotting issue you should be aware of, because if you have too many dots crowded together, you will not be able to see how many dots are actually there. So that's why for plotting, we always encourage you to use transparency so that you can see some areas have more dots than others, right? But because you can see the numbers are totally different by 2,000 versus 11,000. But from this plot, you may not see the numbers to be very different. Okay, and the right one is just to allow you to add the labels of the top 10 genes. So you see the labels here. Yeah, just for your information. But the question I want to ask you is, why do we want to screen for highly variable features? Why don't we use all features? Because intuitively, keeping more information of your data would be beneficial, right? Why do we want to throw away some information? Here's the rationale from, so I think many references will give you similar ideas, but this is just a summary. This is to select genes that contain useful information about biology of the system while removing genes that contain random noise. So I should say I need to update this reference. It should be a different reference, but I'll update that afterwards. And this is what's argued, right? So some genes contain useful information, but other genes measurement variation from cell to cell is mostly random noise. We don't keep them. But I think from a computational perspective or statistical perspective, we want to reduce the curse of dimensionality, because essentially we reduce the number of features from 13,000 to 2,000. And the number of features is the dimensionality, right? In such a high dimensional space, every cell is just 1 point. So therefore, we know that for high dimensional space, we have all sorts of issues, including that the Euclidean distances are no longer so meaningful. So therefore, I think for all these reasons, we prefer to have a smaller dimension if possible. And to do this, to select highly variable genes, which essentially has an assumption that the genuine biological differences are manifested as increased variation. So high variance reflects more likely biological differences than technical differences, while the uninteresting biological variations, say from just the biological noise, transcriptional bursting. So this jargon means that a gene is constantly transcribed. Sometimes its level is higher, sometimes its level is lower, depending on the exact time when the cell is measured, right? But such differences should be considered not so interesting. And also the Poisson sampling difference from your sequencing is not so interesting. So for those, we don't want to keep those genes. But basically, that's what's here. And let me next introduce this VST method in more detail. What does it actually do? This is from the Seurat tutorial again. VST, first fit a line to the relationship of log variance and log mean using local polynomial regression. The short name for this technique is called LOS. Then standardizes the feature values using the observed mean and expected variance given by the fitted line. So given each gene's mean, observed mean across cells, you have an expected variance given by the curve. Then you will call that expected variance. Then feature variance is then calculated on the standardized values after clipping to a maximum. See the clip max parameter. Okay, so we actually have two key parameters in this verifying variable feature function. One is called low S band. This is for the curve fitting. One is called clip max. Let's first look at the low S band. What does it actually do? Yeah, so you see these two things, they have default values. Low S band was set by default as 0.3, and clip max is set as auto. Okay, what does auto mean? After standardization values larger than clip max will be set to clip max, default is auto, which sets this value to the square root of the number of cells. Okay, so essentially what it does, I can just quickly tell you, well, I can do this by some quick writing. So what it does is that for each gene, you would have some count. So basically, let's say gene, I think it uses gene I. So in Seurat, I mean, you follow that. So for each gene I, suppose that you would have the count as Xij of gene I in cell J. Then when you do the standardization, you will extract the, how to say it? You would basically use, okay, so the observed mean is the gene's average across all the cells, right? So I will call that Xi dot bar to mean the average of gene I. And then I will divide it by some square root, the square root of your expected variance. So this expected variance of gene I, I'll just use this notation. Experience variance of gene I is what you get from the low S curve. Then you do the standardization. You call this the standardized value. And then what you do next is the clipping. The clipping means that truncation. So basically for this standardized value, you compare it to some clip max value you previously set. So in this case, the default for auto is that the maximum value is square root of n. N is the number of cells. Then you compare these values to this value. If they are bigger than square root of n, you set the values to square root of n. But I actually have a question for you. Why do we want to do this? Okay, anyway, let's first look at the curve fitting, low S. So this is from just a tutorial on low S from this blog. So low S has this key parameter called span. It's a parameter that controls the degree of smoothing. So you see that unlike linear regression, low S, the curve it fits can be nonlinear. You try to explain the pattern in your data, right? This is your X, this is your Y. But how smooth you want the curve to be, that's controlled by the span parameter. So the intuition is that the larger the span, the smoother the curve. Then my question for you is that now each colored line represents one span. Then which one is the largest one? Which one has the largest span? What's your intuition? Huh? Sorry, what color has the smallest then? So which one has the largest, which one has the smallest? Orange one has the smallest stem. Yeah, because you see this one fits best to the dots, right? And then once you are more smooth, you are farther away from the dots. So actually in statistics, this one is known as the variance bias trade-off. So the intuition behind this term is that when you have the curve more smooth, it's farther away from your data points, and you call that bias. But when we say the smaller variance, so we have larger bias but smaller variance, what we mean is that if you have a different sample of data points, then it's more likely that when you fit low-S, you will get a more similar curve, right? Because the curve is encouraged to be smooth instead of fitting the data points. On the other hand, if you have a very small span, you could essentially go through every dot, then you have no bias. But once you get a different sample, for the new data points, this curve may not fit well. So that's why it's called small variance, large variance. That's what we mean. Okay, so yes, correct answer, orange. So then you can see that here, it has some, influence on your result, because basically when you call this as your mean log, sorry, go back here, log variance, log mean, right? So in Surratt, you would have the y-axis as the log variance, and this is the log mean. Yeah, so you will see that then you are basically predicting the log variance from the log mean using the curve. And then the different curve will give you different expected variance. There is an influence here. And I want to say that there are two other methods in Surratt. One is called mean var plot, MVP. So what it does is that you want to use a function to calculate average expression, mean function, and dispersion function for each feature. Next, so you are basically fitting, how to say it? You are trying to get these two parameter estimates, mean parameter, dispersion parameter, which I'll talk next. So it's actually the dispersion we introduced in the negative binomial distribution, which is actually here. And for each feature. Next, divides features into a number of bins, default 20 bins based on their average expression. So you will group the genes with similar expression values into one bin, right? For the smallest bin, for the one bin on the two bins on the two ends will represent the most low expressed genes and most highly expressed genes. And then you calculate z-scores for dispersion within each bin. All right, so you're taking the dispersion values you calculate for those genes, given the spin, right? Because you're assuming the genes have the same mean. So you no longer look at the mean, you look at their dispersions, and you standardize the dispersions into z-scores. Z-score stands for the value subtracting the mean divided by standard deviation. That's the z-score. The purpose of this is to identify variable features while controlling for the strong relationship between variability and average expression. So you can actually see that it's very similar to low-S conceptually, except that here you're not doing this low-S fitting, but you're basically dividing your X values into 20 bins. Then within each bin, you do the standardization, just like what we do here. Just like what we do here. But here the mean is from this gene itself, right? But over there, the mean is actually by, how to say it? So it's no longer working with the counts, I should say, but you're working with every gene I's dispersion values. And once in the bin, you're assuming that, okay, the dispersion values share a common mean, and then you standardize by the standard deviation. So we're standardizing different things. This is standardizing genes count value, while here we are standardizing the dispersion value. But either approach, we want to remove the effect of mean. That's the similarity. All right, and there's a last method called dispersion. So 3 methods for selecting highly variable genes. VST is the default. The dispersion method selects the genes with the highest dispersion values. So you don't do any standardization. You just select the dispersion values. Then this is a review of dispersion, negative binomial. So in a negative binomial, it's called over dispersion distribution because the variance is bigger than the mean mu because of this additional term, right? This term is non-negative, making the variance greater or equal than the mean. And in this second term, this 1 over r, I don't know if you remember, but this r is from that original formulation of negative binomial. It's the number of failures before you have first success, right? That's the r. So 1 over r is defined to be the dispersion parameter. And this parameter, what happens is that once the dispersion goes down to 0, you degenerate negative binomial to Poisson, right? Mean becomes variance, mean variance equal. When the dispersion is positive and large, you have more over dispersion. So it controls the degree of over dispersion. And the reason why these two selection methods focus on dispersion is because they don't care about the mean. You want to care about the over dispersion degree. All right. However, there's one key thing I want you to be aware of, which is the scale. In the VST method, when we say we fit the relationship between log variance and log mean, what is the scale for our definition of mean and variance? Because in your homework, we talk about the normalization stabilization approach, right? And so in this case, if the data is normalized, then we should expect that log variance no longer depends on the mean. That's something you would check for your homework, the Spearman correlation thing. Then in this case, why would you still need to fit this relationship? That's my question. But if we look into the code of Seurat, we'll find that interestingly, it's for the count data. It's not for the normalized data. So normalized data is not used in this step. And this is found by my student, the reader of this class, Zhiqian. He found that looking at this function, find variable features standard assay, you will see that when the method is VST, counts are used. But when the method is this mean var plot or MVP, you use data. And data contains normalized data from the previous step. So then why do we have this different choice? That's another interesting question for you to think about, right? So whether we should use normalized data from the previous step or we should use counts. These are small issues, but it will affect the result. Okay, so however, if we go to another package, which was written in the old days, and this is a very comprehensive tutorial on bioconductor. Basically, it's about single cell data analysis. It's the RAM package. It's not Seurat, it's not Seurat. So people agree on these steps, but the details are different. In this package, you will see that when we talk about the variance, the mean is the variance of log expression, mean of log expression. So it's whether you first do log or you first do mean variance calculation. That will make a difference because here you are doing log variance, log mean. Here, variance and mean are based on counts, right? While here, you are doing variance of log, mean of log. So which one do first? That will also affect how you define highly variable genes. But in the SURAM package, the highly variable genes we want to pick out are those. Basically, they are the genes whose variance are clearly big given their mean. So the intuition is the same, but the implementation is different. Okay, so I think I don't know how large the difference would be. Honestly, the reason is that the number of highly variable features we select is kind of large, right? It's 2,000 instead of 200. So therefore, maybe given that large number, the differences will be minor. But whether that's justified or not, that's another question. How do we come up with that number? Is it based on some empirical evidence? But how would we decide on a different number if our data set is special? I think these questions are all given to the analyst. Yeah. But I want you to be aware of these parameters here. Otherwise, you would just go for the default. Okay, and there was an old review paper, 2015. At that time, single cell was still a very new technology about the challenges. And there was one section about identifying highly variable genes. So let's look at the perspectives here. It offers a different perspective. So in parallel to differential expression analysis, an important challenge is to identify the genes with the most highly variable expression patterns across a population of cells without prior knowledge of the underlying cell types. Gene expression variability can provide clues regarding transcriptional heterogeneity in the sample of interest, given insight into the robustness of gene expression regulation between cells, as well as cell type characterization. The identification of highly variable genes requires the application of statistical approaches that account for technical sources of variation, such that biological variability in gene expression levels can be quantified. In addition, it is important to realize that high variability of gene expression can also be caused by a confounding factor that's not accounted for, such as cell cycle. So this paper mentions an important thing, which is the consideration of confounding factors, whether their effects on your variance is what you're interested in, or we should remove them, right? If you want to remove them, what would you do? So very soon we'll show you that you could remove them in the regression approach by directly doing that in SC transform, okay, because it's a regression-based framework. And this is following this paragraph. It's just an excerpt from this review article. One approach is to compute the coefficient of variation, that is, the empirical variance divided by the square mean for each gene across the population of cells under study, and to rank the genes accordingly. Unfortunately, technical variability, which is intrinsic to the experimental protocol and not associated with genuine biological variability, is greater for lowly expressed genes than for highly expressed genes. Consequently, a null estimate of the expected technical variability needs to be computed. This can be done using the extrinsic spiking molecules. The extent of variability in their expression across cells can be used as an estimate of the null variance. This information allows the expected technical variation to be modeled across the whole dynamic range of expression, which forms the basis of a statistical test to determine the set of genes that show more variability in expression than would be expected by chance. Recently, extrinsic spiking molecules have been used to further decompose technical variability into two terms that correspond to sampling noise and heterogeneity in sequencing efficiency across cells. So as this paper was from 2015, I want to add that for now, if you use the 10x genomics protocol for your sequencing for single cell data, you wouldn't have extrinsic controls. You wouldn't have this extrinsic spiking. I think I might have briefly mentioned this in a previous lecture. That is, you can add RNA molecules with no concentration to your solution, and they can be encapsulated into your droplets, but they do not represent the RNA in the cells because you have this cell lysing step, right? That kind of variability cannot be captured by the extrinsic spiking. And I think that's the ultimate challenge here. So the spiking may not be that realistic, unlike the bulk data. Because in the bulk data, you would extract the RNA, add a spiking, right? So then the RNA from the cells, from the sample, biological sample, they are already in the solution. But in single cells, it's not the same procedure. But this coefficient of variation, I want to make a note here. This definition here is not accurate. It says that is the empirical variance divided by the square mean, which should be standard deviation divided by the mean. So it's not variance divided by squared mean, but it's the standard deviation by mean. So the term introduced here is the square of this definition, basically. It should not be squared. But this coefficient of variation, you see what it really does is that when you divide standard deviation by the mean, so these two parameters should be on the same scale, right? Their unit should be the unit of your data. For example, if your data are measured in RNA copy number, then the standard deviation is the standard deviation the copy number, and the mean is the mean copy numbers. They're on the same scale. So when you do the division, you remove the unit. Right. On the other hand, you're also trying to say that, okay, when I talk about standard deviation, I want to remove the effect of the mean. So this is it. But you can see that whether you want to do this division, or you want to do subtraction. You can do something minus, right? Standard deviation minus the factor mean. And that's what the regression framework is doing. When we do the log variance log mean, we're removing the effect of the log mean. So I think these are all tricky issues in bioinformatics. Which scale do we work on? The original scale or log scale, right? Then which way do we use to remove the effect of one thing on the other? Do we do division or do we do subtraction? And here, I think when people talk about coefficient variation, I think they're talking about the original scale, not the log scale. But you can see that the division on the original scale is equivalent to the subtraction on the log scale, right? Because log, inside log, if you have x over y, then it's equal to log x minus log y. Yeah, so you have that fact there. But these are the things I want everybody to be aware of. Yeah. So basically, this is an overview paper. So it's not the same as now, but I think these two things are worth noting. One is confounding factor. The other is how do you remove the effective mean? Then next step, after we select the highly variable features, say default 2000, we do scaling. Okay. So when we scale the data, what we actually mean is kind of like z-score. We try to set the mean to be 0. We try to set the standard deviation to be 1. So, and the hope is that we want when we want to run the PCA, we want to make sure that all features can have equal influence on the PCA without any prior, bias. By bias, I mean that if we have some genes with larger variance, then they will, of course, dominate the PCA result, because PCA is actually a technique which we'll introduce next lecture, is trying to explain the variance in your data using fewer orthogonal dimensions. So you want the pieces to be orthogonal to each other, and you want the first PC to explain the most variance in your data, and the second PC to explain most of the remaining variance in your data. So then the variances explained by each PC, since the PCs are orthogonal, can add up to be equal to the total variance measured by genes, right? So previously, you will have one variance per gene. If you add them up, you get a total variance. And then when you do PCA, you're basically distributing this total variance to be explained by PCs, and they are orthogonal. So you can imagine if we don't do scaling, then the genes with larger variance will play heavier roles in the PC. But when we standardize them to have the same standard deviation, then the PC will be more driven by gene-gene correlation. So that's something we'll show that later. Basically, you'll see that for genes more highly correlated, they will be explained by one PC. And this PC can be interpreted as some gene group. So I think these are the reasons why we do the scaling. So what do we do exactly is that we shift expression of each gene so that the mean expression across cells is 0. We scale the expression of each gene so that the variance across cells is 1. So this step gives equal weight in downstream analysis so that highly expressed genes do not dominate. Ultimately, PCA is developed as a method for normal-like data, like multivariate normal data or Gaussian data. So we want to, again, turn the data more Gaussian-like. And this is actually achieved by the normalization step, actually, by the variance stabilization thing. So make the variance no longer depending on the mean. But then before we supply the normalized data to the PCA, we add the scaling step so every feature on the normalized data scale have the same standard deviation. All right, and the results here are stored in this scale data thing. By default, only variable features are scaled. That's from the previous steps. So if you have 2000 highly variable features, you only scale them. We are not scaling the rest. Yeah, but since the scaling here is done one feature at a time, it actually doesn't matter, right? Because when you scale more features, the top 2000 feature scaling wouldn't be affected. All right, but you can specify the features to scale additional features. Basically, you have this argument, features, right? If you don't put anything here, by default, only the highly variable features are scaled. Otherwise, if you put all genes, then all genes are selected, are scaled. So it's something trivial, but I think maybe if you scale fewer genes, you will save some storage for the scale data. Yeah. Okay, but there's a note here. How can I remove unwanted sources of variation? Yes. Yes. Yeah, you only have the 2,000 genes in scale data. Yeah. And for this removal of unwanted sources variation, this is actually about this thing. What if we have a confounding factor that's not accounted for and that will affect gene variance? Okay. So in Seurat, we can also use scale data function to remove unwanted sources of variation from a single cell data set. For example, we could regress out heterogeneity associated with cell cycle stage or mitochondrial contamination. Right, so you have this argument in scale data called vars to regress, variables to regress out. Here, the example was given to remove the percent mitochondria. Do you remember this, right? This is what we use for filtering in the first step. We think if the percent mitochondria is too large, then it is implying that the cells are dead, right? Then we don't want to keep those cells. But for the remaining cells, maybe their percentages of mitochondria still vary. They're not equal to each other. So therefore, you might want to regress it out so that it's no longer affecting your data. So scale data, you have this option. But this author said, particularly for advanced users who want to use it, because the default here doesn't regress out any variables. You have to specify yourself here as an advanced user. The authors of Seurat strongly recommend users to use acid transform. And so basically, acid transform uses the negative binomial generalized linear model. Then you could just include your confounders there for removal. Okay, but the thing I want to say here is that, how to say it? Since we have seen that other parties' paper doing this benchmarking or analysis of normalization method, which found that acid transform doesn't have the nice property of stabilizing the variance, that leaves another question here, right? Which one should we use? So just to quickly summarize the issues we have encountered, oh, maybe I should move on to this. So this is just, I'm saying that in the regression model of acid transform, if you want to remove confounders, you just add them here. So then when you get the Pearson residuals, if you remember that term, as your normalized data, you have already removed your confounder effect. That's basically it. But I want to say here are several issues we should be aware of. First, what's the standard of normalization? Whether there are confounders we want to remove, right? And when we define highly variable genes, what is our criteria? So for all these issues, these issues, they would affect the data we input into PCA. And then why do we need PCA? PCA is basically the foundation of almost all the key analysis you will do. So for example, if you want to define cell clusters, you first do PCA. If you want to visualize cells in 2 dimensions using t-SNE or UMAP, you do PCA. So, but all these steps we talk over here, we talked so far are about the input into PCA. So I will say that even though this is the state-of-the-art single-cell analysis pipeline, I think something which is more challenging than before is that we have more steps than before, right? Compared to both data analysis, we added more steps, and their effect can be quite large, but still depend on the data set. So for example, the differences of the analysis results depend on the package. For example, Seurat, ScanPy, or this older SU1 package. How do we explain those? Which one do we trust? I think for all data analysis we do, we need to be aware of this question. Yeah. So yeah, actually, I think I went a little fast today, so that's the thing I have prepared for today. But before we continue, I think I can go into the next topic maybe earlier than expected, but I want to take the time to ask for your questions before we move on. Yes. Like for here, they want to remove the percentage of mitochondria. And it also mentioned the cell cycle stage. If that's something you don't care about, like the cell cycle stage each cell is in, then you could maybe add a cell cycle marker genes as your confounder. So then the variance is not depending on them. But I have to be honest that I haven't seen many analysis doing this confounder removal, because the more things you do to change your data, there's a risk that you lose some information. And again, that's an arbitrary choice, right? But I think maybe people's approach is more like, okay, I keep the default analysis, but if I know there's something wrong, then I consider whether there are some confounders I might need to remove. Yeah. So again, you can see that analysis is very tricky, right? It's depending on the people who do the analysis. So that's why I think, at least for now, maybe the only feasible way to make our results transparent is to record the detailed steps you do the analysis, right? Every step, what did you filter out? What transformation did you do? And it's very important to keep that record, just like the experimental record for your notebook, right? When you do experiments, so you know what did you change or whether something might cause the issue. Yeah, that's the only way out for now. But I mean, as a computational person, I will be very interested in seeing whether it's feasible for us to do the analysis with fewer steps. Yeah, instead of step by step, because you can see there will be a cumulative effect. Okay, yes. Very good idea. Statistically, I would prefer the lowest. Yeah. Because for binning, it's first of all, people call this hard thresholding, right? By hard thresholding is that once you define the bin number, you are only looking at the genes within the bin, but not the neighboring bin, right? Then the number of bins is critical, right? But for lowest, you would actually, like this is a low S curve, you would actually use this all neighboring genes, but their degree of influence on where the curve is at this point depends on their distances in the x-axis. So the closer points have bigger influence, farther away than the points have smaller influence. So it's more like gradual, right? But binning is like you just set a boundary and you only look at the boundary. Yeah. Sure. So is the max yeah. I'm sorry, could you clarify what you mean? Because, okay, so let's just use this as one example, even though it's not exact. But what I mean here is that, so once they have the fitted curve, then for each dot here, they will take the Y value, right? So basically this Y value will be changed to the value on the blue curve. That will be your expected variance. Then for each gene I's count, you will subtract gene I's average mean across cells divided by the square root of the variance on the blue curve. Then that's your standardized value. Then you clip on the value. Yeah. Yeah. So the way that I think of it seems to me that I think there's not a lot. The maximum value is exactly. Okay, maybe it's better to go back to this figure. That's closer to what's happening right here. By clipping, right? Basically, you essentially, okay, it's not exact here, saying, because this is the standardized variance. By standardized variance, we're making the calculation of variance after the standardization, after clipping. So using those two steps, then you get the clipped standardized gene expression counts. And then you calculate the variance. That's called standardized variance. That's what you get here. But essentially, the clipping is not happening on the standardized variance, but happening on the standardized values. Yeah, but the intuition is right. We don't want the values to be too large, then they will dominate the variance. But my question, or I don't understand, is why do we set the default clipping max value to root n? That's the part we don't understand. If you have more cells, you want to be more permissive. That's what it does. But what's the rationale of using root n? Yeah, actually, the way I thought about it was that we know something is root n scale in statistics, which is the standard deviation of the sum of n samples. So like if you have x1 to xn, they share the same standard deviation, then the standard deviation of the sum of x1 to xn is square root n times the standard deviation of each one. So that's where the root n is. But I don't see the connection here because it basically says that the larger sample size, the sum's standard deviation will increase on the order of root n. Yeah, but here we're not looking at the sum, we're doing outlier detection, right? We're basically doing outlier detection. Why do we define the outliers to be, okay, why do we define outliers based on sample size? That's my question. And it's also the same root n thing is used in SC transform for removing outliers. but you could have n. Yeah. Yeah, I don't understand. I should say I don't understand for now. That is an open question. So there are many open questions, especially outlier removal itself is always a difficult question. It's always a question, yeah. But people can use something like called robust, the robust statistics. So by robust statistics, we mean that the median is more robust than the mean, right? You could have the robustized version of the variance. So yeah, so there are multiple ways to do it, but that doesn't require to remove anything. Yeah. Just give me some, yeah. Yeah, please continue. I think there counts. That counts. At least when they fit this relationship is the true counts. Yeah. And it didn't say whether the standardized the feature values is on the log scale. I don't think so. Yeah. Because but we have to go to like details to check these. But this is the variable sub, this is the VST method, right? So method VST. And yeah, I think it's on the count level. Observe mean, observe variance on log scale. Yes, log variance versus log mean, then you do lowest. And then basically once you get expected variance given the fitted line. Yeah, that's a very good question. You need to go back to the original scale, right? Or you want to standardize the log scale values and then, yeah, good question. Well, I think that's why I think we need more detail here. Yeah, it was exactly being done. Especially you have the normalization step before it. Yeah, very good question. Yeah. I don't know how many users are really, checking these options. I highly doubt it. I think people may go with the default. Given that you have so many parameters here, yeah, like I said, maybe people will only go back to check the parameters if you notice anything unreasonable. Yeah. But one thing I want you to be aware is that there's some discussion about the differences between Surat and ScanPy and on Twitter. And I think that there's a very interesting message, but I didn't really confirm myself. I will share it with you just for your check. So the message is that if you want to have Surat and ScanPy to give you consistent results, you have to downsample your cells. Maybe you have to just filter out those outlier interesting cells. And for the core small set of cells, then their results are more consistent. But the more cells you include, their differences will diverge. So basically, the larger sample size will drive bigger differences between the packaging. But this is very counterintuitive to science, because we want to measure more cells for sure, right? That's why we go with the high throughput technologies, and we like the dropseq versus the plate-based single cell, because using dropseq droplets, we can measure more cells. That's our goal. But if that will drive bigger differences between packages, which one would we choose? Yeah. Yeah. Yes. Yeah. So you already have the versions of these functions in ScanPy. So the reason we didn't make it just restricted to R, because we want to leave the open option for people who don't know R at all. That's just the only reason. Yeah. Yeah. I think some technical details, like the code implementation. So like I said, even though the high level ideas are similar, but how do you actually define highly variable genes? It could be different, right? So how do you do the normalization exactly? The default, I think the default in ScanPy is not log1p CP10K. I think that's another reason. Yeah, maybe the defaults are not the same. And even though they look the same, the detailed implementation may have some subtle differences. Yeah. Lecture notes, they are all on Bruno. I post it after every lecture. I will try, but as this is like a new lecture, it's not something I've taught before. I mean, I'm not using the old material, so that's why I need some time to prepare, but I will try. Yeah, it's not like, because I have to make the slides the day before. But I could, of course, push my schedule a little early, but just for your understanding, you get busier as you become a senior faculty. You have something like the lab to manage, but I'll try to be earlier. Yeah, but still, even after that, I may still make modifications right after lecture. So the version could be updated. So you're basically laughing. Yeah. No, it's not an excuse, but if you check people, just for your information, I think it's a fact, I can say that. If you check the PIs who run a lab with the same size as my lab, maybe I do the most teaching. Yeah. Yeah. No, I'm just trying to explain why I might not be able to do it, but I'll try. Yeah. Even this new lecture, because otherwise I could use my old materials, right, to teach the same class again. Yeah. But I think for this new materials, I do feel, wow, it's more challenging to do the single cell data analysis. There are more and more steps added. And also, if you go from thread 3 to thread 4 to thread 5, you will always have some changes. And how many people will actually go to, I mean, this level of detail checking, right? I'm actually very impressed by my student who did the check. But on the other hand, it's not something you can expect from users. Yeah. But okay, and let me move on to the question I want to share with you. This is a question I have myself after going through the steps. I'm saying that here the question is, why is normalization based on all features instead of highly variable features if only highly variable features represent biological variants? Right, because the rationale for doing the highly variable feature selection is that they represent the biological variants. The remaining features do not represent biological variants. If that's the rationale, why would the normalization you do based on all features? Because when you normalize the size factor, cell library size to 10K, say, you're using all genes, right? Yeah, I actually have this question myself. Because it's not like, especially the default VST uses counts instead of normalized data, right? So basically you go from the standard pre-processing, this is the cell filtering, you go to normalization, you go to identification of highly variable genes. It's not like sequential between these two steps. But then why would normalization be using all genes? Yeah, anyway, I think normalization itself is something so tricky, right? To justify. And again, another thing I want to share with you, I think, is that normalization, the need for normalization is not the same for single cell data and bulk data. The key difference is that for bulk data, you measure samples separately. You're running on different sequencing experiments. But for single cell data, the cells are measured together. So therefore, to me, it shouldn't be considered the same problem. For bulk data, I think it makes sense to normalize the total library size to be equal because you don't have absolute values that are comparable. You can only compare relative values of genes within the sample, right? Then basically doing this total library size normalization is to let you look at the relative expression instead of the absolute expression because you don't have that information anyway. Since every sample can have different sample capture efficiency, PCR may do different things on different samples, and these will all make the sample's total library size be different. And also, you may have different amount of biological material in different samples. So, right, so I think for bulk, it makes sense. But for single cell, I think I mentioned this previously, why do we think the cells should have the same library size? Some cells are bigger than others. Yeah, so that's why I still feel like it's a different problem. But for single cell data, probably the need for normalization is mostly for visualization, clustering. These are kind of depending on Gaussian-like data. Yeah. Yeah. Sure. We have a bunch of hallmarks. Uh-huh. These are based on for scaling. For the normalization. Biological, okay. But then would you say that you believe in the normalized data more than the count data after normalization? In what way? Right? For what tasks would you believe in more about normalization data, normalized data? Because my question is, if you believe in your normalized data more, why don't you use normalized data for highly variable gene selections? Yeah. Yeah. I know for some statisticians' preferences, they prefer to work with count data instead of normalized data. So if you need to adjust for anything like cell library size, confounders like percentage mitochondria or cell cycle genes, you include them into your count regression framework instead of doing the normalization first. But the challenge is that when you work with count data, any model for that is computationally more intensive to fit than Gaussian models. Yeah. Yes. Order of columns, you mean cells. Columns are cells. Rows are genes. So for the log 10K normalization, sorry, let's go back. For this normalization, you do not keep, if you look at for the same I, gene I, you look, sorry, it's gene J, okay. If you look at for same gene J, you look across cell I's, then the relative order of different xij and xi prime j, so or x1j, x2j for two cells, the relative order may not be kept after normalization because the two cells may have very different library size, right? Then after you divide the library size, one could become bigger than the other, even though it's smaller before. So you would change the relative order of the same gene across cells. That's the effect of normalization. It's not like monotone transformation. And that's exactly, I think, what people want to do normalization. I mean, at least for the bulk data, right? So for bulk data, it could be this gene has a count 10 in sample 1, has a count 100 in sample 2. But if the total sequencing depth between the two samples, like sample 1's total sequencing depth is fewer than 1/10 of the second sample, then you may say, okay, this gene 1 is not so low expressed in sample 1 because sample 1 just have fewer reads. I think for that, bulk data is obvious, but for single cell data, it could be that, okay, cell 1 is just very small, have very small amount of RNA. Then gene 1 indeed have a smaller amount of RNA compared to cell 2. But the relative proportion of this gene in the two cells may be the other way, right? So I think ultimately it's always about whether you care about relative expression or absolute expression. Yeah, I think the difference for single cell data is that since the cells are measured together, then you could talk about absolute differences. But for bulk data, you cannot talk about it. Yeah. Okay, anyway, so I think just to let you know that even though the technology has been there for, I mean, at least for 10x, has been there for 6 years, and the CRAP pipeline has been mature, right? Kind of mature, version 5. Many people, at least on the software engineering perspective, is very stable and very efficient. But we still have many things we don't understand from analysis perspective. Yeah. Yeah, and how do we deal with them, or we just embrace them, embrace the complexity, but be aware of the issue that might happen in our data analysis, or we can think about simpler solutions. I think these are all open directions. Yeah. Yeah, but sometimes it depends on the project you work on. For example, if it's an analysis project, right, you have to embrace the complexity and get the signal you care about. Probably the signal, if it's strong enough, then you should find it no matter, or if you change the tuning parameter, you should still be able to find it. But if you are a method person, it really depends on, sometimes it's your taste of what a method should be like, right? Whether simplicity is your preference or you don't care about complexity, or yeah, I think that becomes more subjective, more arbitrary. Okay. All right, so then maybe we can stop here, and then next lecture we'll talk about PCA. Yeah. Thank you.